Great. Well, good evening and welcome to the Florey for this special event for World Epilepsy Day, or Purple Day as it's known in the community. I'm Professor Peter van Wijngaarden, Executive Director of the Florey, and I'm delighted to see you here in the audience. Among us we have committed donors, supporters, advocates and research collaborators. I understand that we're also joined online by a large number of people who are participating as part of our live stream, so I extend my welcome to you online. So before I begin, I'd like to acknowledge the Wurundjeri people of the Kulin Nations, the traditional owners of the lands on which we're meeting. I pay my respects to their elders past, present and emerging, and I extend that respect to any Aboriginal and Torres Strait Islander people we have in the audience today. So as you may know, epilepsy is a major focus of research here at the Florey, and it's one of our greatest strengths. Research here at the Florey spans from improved detection and diagnosis of epilepsy to understanding the genetic and molecular underpinnings of various forms of the disease. Several of our researchers are really at the forefront of developing promising new treatments for epilepsy, and members of the Australian Epilepsy Project team are setting the new standard of care for the diagnosis of epilepsy using state-of-the-art imaging, as well as artificial intelligence tools and telehealth technology to assist neurologists in the diagnosis of epilepsy. So we're going to hear this evening about several aspects of this research. Now, if there's one thing that scientists and clinicians are renowned for, it's jargon. And this evening, you're going to be hearing a bit of jargon. For instance, you're going to hear about the DEEs, developmental and epileptic encephalopathies, otherwise known as severe childhood epilepsy, genetic epilepsy, or early life epilepsy. These terms all refer to a group of epileptic conditions that typically have their onset in early life and are often accompanied by developmental delay, autism, intellectual disability and movement disorders. Now, many DEEs are named after the genes that are associated with them. Examples that we'll hear about this evening include SLC6A1, HCN1, SCN2A and PURA. Now, as complex as it is to talk about these conditions, the reality for patients and their families, from navigating the healthcare system for a diagnosis through to finding suitable treatments, is even more complex. Fortunately, we have a superb lineup of speakers this evening who are ideally equipped to guide us through these complexities. So gracing the stage tonight will be Florey researchers, Professor Reid and Associate Professor Snezana Maljevic, and together with Paediatric neurologist Katherine Howell. We'll also be joined by two very special guests, Mr. Stuart Place and Mrs. Mel Anderson, who have children with genetic epilepsy. And Stu and Mel will share their experiences with us in a panel discussion. It's now my great pleasure to introduce the first of our speakers, my friend and colleague, Professor Reid. Chris is head of the epilepsy mission at the Florey, and he runs the Neurophysiology of Excitable Networks laboratory. Come on up, Chris. His team investigates the molecular and cellular causes of epilepsy, and Chris has devoted his career to making a difference in the lives of people affected by epilepsy. So please put your hands together to welcome Chris.
A little taller. Welcome, and thank you, Georgia, for giving us the opportunity to talk to you today. My job really is to convince you guys that we've got a paradigm here at the Florey that's changing lives, and perhaps to convince you further that we need to increase that capacity so that we can help more kids with this devastating disease. The core of the talk will be precision care for the children with developmental epileptic encephalopathies, or DEE for short, which is much, much easier. And a lot of what you'll see today, is, at least from me, is around the mouse, and that'll become clear. But first, before we start, I'd like to set a video. This is a little girl called Fern who has HCN1 DEE, her dad talking about the condition and the impact that it's had on their family, and the impact that Florey has had as well.
We all envision how our life's supposed to go, and when something like this happens, all that changes. It's like being struck by lightning, there's no explanation, it just happens. Jules was bushwalking and Fern just went totally floppy and face went blue and stopped breathing. I'd say that's probably still her most traumatic seizure, hundreds of seizures later, because of the unknown. About 2, I think, is when she started having episodes where after a tonic-clonic seizure she would then collapse and stop breathing and go sort of blue from head to foot. At that point, seizures were still very confronting and hard to look at and hard to deal with and scary. We were trying different medications, not having much luck, lots of loops of hospital and ambulances, and eventually we got some genetic results and figured out that she had an HCN1 gene mutation causing her epilepsy. This is, as an individual gene, extremely, extremely rare. I think there's 4 or 5 Google results and a lot of them have the Florey or Chris or Lauren's name on the studies.
The Stevens family actually got in contact with me late last year. We talked through where we're at in terms of understanding HCN1 epilepsy. We also identified that one of the medications that Fern was currently being treated with at the time is something that in our studies in the lab and also with a lot of anecdotal evidence from other patients typically makes things worse.
Taking her off that one that was exacerbating things had a really immediate result. She had a really bad tremor in her hands that just disappeared overnight. Socially, she's better. We're 12 weeks seizure-free and counting. If we can keep the research going, it's so exciting that it's in Australia and at the Florey. So if we can keep that going, there's potential to help a lot of kids. It just disappeared overnight. Socially, she's better. We're 12 weeks seizure-free and counting. If we can keep the research going, it's so exciting that it's in Australia and at the Florey. So if we can keep that going, there's potential to help a lot of kids.
It's always been told to speak up because I'm so tall. It's always very difficult to follow that. And in some ways it's stolen the thunder, but I think there's no better person to do that than Phil. Very dry slide, and Katherine's going to do a much better job of describing the clinical aspects towards the end of the talk. But I'd like to make a couple of points here, importantly, that DE occurs in about 1 in 590 births. So the individual causes that Fern and Ebony, who you'll meet, and Sarah, etc., are relatively rare. But collectively, the disease occurs in about 1 in 590 children per birth, and that equates to about 500 children that have the disease here in Australia, and obviously you can amplify that across the globe. The other really important point here on this slide is that we can now get a genetic diagnosis. Again, Katherine will talk a little bit more about that. But what that means is that we've got the genetic start point. We've got the starting point of what causes these devastating conditions, and that's been incredibly important for moving this field forward, in part through the stuff that I've done, or collectively have done with mice modelling, but also, as Snezana will talk about, also stem cell or patient-based stem cell modelling as well. So that's the dry slide, but obviously D is much more than just a number. It is what we've just seen, children with this devastating disease. And for this story, I want to introduce you to 3. I mean, Fern, you've already been introduced to, but also to Ebony. And you've met Lauren already, but also Chasely and me. Important slide because it is telling us that it's not just me, it's a group effort that's behind what we're doing or what I'm going to show you today. And lastly, Sarah. Mel will be part of the panel, and she'll describe some of the issues that have been part of her life over the last 16 years. Is that right? Yeah. So again, it's not just a number. It's much more than that. And the story, at least my story in this field, mostly began with Ebony. She's a little girl in Adelaide who has an HCN1 variant. Her dad approached me, what must be almost 6 years ago now, having had the diagnosis. It was very new, nobody had seen it previously, and asked, well, what can we do? I had been working in the HCN field prior to that. That was the reason that Tim got in contact with me. And we decided to make the mouse, and you'll hear that as we go through. But this is one of the first emails that I got from Tim. It was around Christmas time, and I fired an email saying, how are things going? And he goes, well, Ebony's going okay, but I think you guys can read that that's not entirely the case. What this highlights is the times that the seizures happen and how nasty they are. For tonic-clonic seizures is the TC there, 4 a.m, I think we all know what that is, in the middle of a thunderstorm, so, pretty tough times. But it also highlights another problem with this disease, and that is that the developmental delay. The seizures are one thing, but these children often, or invariably, have quite severe developmental delays, and that's something that often scares parents even more. I'm not convinced that we've got an answer to that yet, but at least with the seizures, I think we're getting there, and hopefully I can convince you of that. Phil's amazing in the way that he articulates, but he also writes very well, and to me this was very impactful as part of something that he wrote for the Florey. The impact of having a child with these types of seizures or epilepsies or omnipresent spectre pervades every aspect of a family's life. It is in the truest sense life-changing. Confronting relentless seizures, constant lengthy hospital admissions, sleep deprivation, etc., etc. Mel will tell this much more clearly than I can. But one of the big issues with these patients when they first arrive and they get first diagnosed is that nobody actually knows what drug that they should go on. And I've termed it medical roulette. That can sometimes cause a little bit of friction, but essentially it is. The neurologist has a sort of idea, and it's getting better. Oops, sorry. It's getting better, there's no doubt about that. But for the most part, we don't know what drug an individual patient needs to go on from the beginning. And it took Sarah up to 20 drugs, and it was still not completely controlled in terms of seizures. But again, I think Mel can tell that story much more compellingly than I can. So how can we help? Well, the genetic diagnosis has become almost 50% of these children. We know the starting point, and it's amazing how things converge over periods of time. So there's a whole lot of new technologies that were coming online about 10 years ago now, where we could use what Peter's termed molecular scissors to put the genetic variant that Ebony has into the mouse. And what that means is that this mouse essentially has the same disease-causing genetic change as Ebony does. This is a mouse that we termed Ebony, the Ebony mouse, for obvious reasons, but the story behind that is Ali came one day and had a look at the mouse and said, oh, Ebony mouse, and I don't think we would have called it an Ebony mouse otherwise, but the name stuck, as you could imagine, and it's a mouse that we've come to love in many ways. So how has it been useful? Well, it turns out the story I've just been talking about, this medical roulette, where you've just got no idea what drugs to put these DE patients on. So Ebony was started on a drug called lamotrigine. Lamotrigine is what we call a sodium channel blocker. The details of that are not so important. But what was interesting was when she was placed on this drug, and this is a seizure score that her dad did on the left, or at least my left. And what you can see as the dose got higher, her seizures got worse and worse and worse with time. But you don't know at that point whether that's just what's happening with these children, because they often have seizures that are continually going, or is it the drug that's making things worse? And this is where the Ebony mouse came in. So we gave the Ebony mouse a whole lot of standard of care drugs. These are the drugs that any doctor can write a script and go to the pharmacy to, that are used to treat epilepsy. And what we found almost immediately was that lamotrigine increased the seizures. So that mouse was having seizures. And what that told us immediately was that lamotrigine was probably having an impact on Ebony. She was likely making seizures worse. And it's completely counterintuitive. Like if I gave everyone in this room lamotrigine, most of you would actually have a lower seizure threshold. It wouldn't have have the opposite impact. But in some patients, it actually makes things worse. And knowing what things make things worse is as important as knowing what works. And I think Fern's story describes that pretty well as well. So it is life-changing. So this is Ali and Ebony on a round Australian trip. I've had a part of the journey. I don't think I know the whole journey. I can't. But they had to buy a caravan that was big enough to have oxygen-making facilities on it, because They couldn't carry enough oxygen bottles for her nightly needs. So I think that highlights it. But the one thing that we did do is we managed to get her on a series of drugs that at least controlled the seizures, which allowed this journey to occur. So that was, obviously an amazing impact already. The impact is not just in Australia. These aren't just children that are in Australia, they're worldwide. And this is an example from the HN Warriors Facebook. Again, you can see the parent from Poland saying, "Help me," the absolute fear of having a child with having seizures up to 3 days tonic-clonic. But then from our perspective, the thing that was really quite nice was this is a South African girl that was on carbamazepine, which is another sodium channel blocker. She was removed and she has been seizure-free since that, as to the best of our knowledge. And that was on the back of the work, the ebony mouse work. The Fern story you've already heard, and that really was an overnight improvement from around 10 seizures a month to more or less being seizure-free. It's not perfect, but it's much, much better than where she was before. And it's sort of nice. It's like to pinch ourselves to sometimes believe it. So. And that was, obviously a conversation that Lauren and I had with Fern's parents. All right, so this is a complicated slide, but the concept's not so complicated. If my idea is right, not every mouse with a genetic mutant in it should respond in the same way to every drug. Otherwise, it's a mouse thing, not a human thing. So we've generated another series of mice, and all of these mice are based on human mutations, so patients that are in here in Australia. And what I hope you can appreciate is that the spectrum of the drugs is different. This is probably highlighting it a little bit better. So the valproate, HN1, that's the ebony mouse, you can see it improves things, and we know that works in ebony. Lamotrigine, HN1 makes things worse. What's interesting is the CUX2, it doesn't seem to work. So you would argue, and we don't know this yet, we don't know from clinical studies, but you would argue that maybe lamotrigine wouldn't be a spectacular drug in the context of CUX2. And in fact, in GABA-B might make things, the GABA-D might make things actually slightly worse. We need to test that, and that's part of what we're doing with Katherine. I think with the ebony case, we can be much more certain. But what I hope I can highlight here is that we can make mice for these individual patients. We're going to have an opportunity to potentially find out what drugs they should be on right from the beginning of the journey. And this is where Mel and Sarah come in. We have now got the Pura mouse here. Again, Mel will talk a little bit more about this. But the hope is that we can do a similar sort of paradigm and get a much better understanding of what what we call pharmacosensitivity of this mouse is, in a hope that we can at least get Sarah and 30 more patients, or is it more now?
Just over 30 in Australia, and we've got 750 globally.
Yeah, so this individual mouse is not going to just help Sarah, but it'll help many, many more. This is me doing the 2-kilometre walk, not run. Although this year I have committed to 10 kilometres, and Peter's going to do the marathon, or at least a half marathon, I think, we hope. And that'll be in support of the Pura work. This is the vision, I hope I've already articulated. We want to make as many mice as we possibly can. And we have been shortlisted for a Frontiers grant in this. It is an expensive exercise, but I guess our hope is that we can help as many children as we possibly can. And of course, it wasn't just me that did all the work. I have a fantastic lab, so thank you to them as well. And I guess the questions will be held towards the end, and I'll hand it back to...
Thanks, Chris. Really excellent talk and fantastic work. We talk in some other areas of science about creating digital twins of ourselves, using our imaging, our genetics, our pathology, and getting the computer to analyse individuals. And what your group is doing is creating a mouse twin so that we can really understand the complex biology behind these conditions. So really is groundbreaking work, and Chris is very modest, but being shortlisted for this Frontiers grant is an extremely prestigious and highly competitive thing. So we're all hoping that you're successful in that endeavour. So it's now my great pleasure to introduce Associate Professor Snezana Maljevic. Snezana is Head of Epilepsy Functional Genomics Group, and she's going to inform us about some emerging treatments for epilepsy. She'll treat us to an overview of her work, particularly in the field of gene therapy. So please give Snezana a rousing applause.
Good evening, everyone. Thanks for being here. I just have to, yes. I will actually talk about the next layer of precision medicine. So continuing from where Chris started, introducing how we can find a therapy that works in the best way in a patient now. We are going to look into the possibility to treat the root cause of disease. So to go after that gene and that genetic change. For this, I need to explain two concepts. One is the patient-derived models, and the other one is gene therapies. So, Chris already mentioned more than 50% of this type of epilepsy have been linked to mutation in one of the more than 1,000 genes at this stage. And what that means is that this DNA that we see in a cartoon will actually have a mistake in there. So there'll be a simple misspelled letter. We know that DNA is a mastermind in the cell. It sits there and governs the whole cell. So in order to produce proteins that are going to run our cells and do all the work, they need messengers. So this is the RNA or messenger RNA. And that mutation that we see in DNA will also be found in the RNA and will cause a protein to look different. So how about we think of this protein as simply a potato? If you think of what DE mutations can do to the protein, they can either make it work less and be kind of a couch potato, or make it work more and be an overachiever. And this concept is important because for us to target genetic or to introduce gene therapies, and especially the one that I'm going to talk about, we need to know what is the impact of a DE variant we are studying. So, in order to introduce gene therapy, we can go back to DNA and we can try to fix that wrong letter, but those molecular scissors are still not approved and so easy to use in human brain. What we are doing, or what we can do, and where the therapies are developing really quickly in DE, is to target the RNA molecules, so to target the messenger and fix that level. So antisense oligonucleotides, that's the name of the therapy. So that's the therapy that goes for the messenger RNA. That one is actually based on a short DNA molecules. They are kind of modified, they can survive everything in the cell, but what they do is that they bind really specifically, so they recognize the letters in RNA and can bind to RNA. And on your left, you can see in a case of an overachieving protein that works too much, we can actually use an ASO that binds to RNA and prevents it to produce protein. On the other side, for a less active protein, so for the one that doesn't function well, we can again use ASO and change either the way the protein is, the levels of the protein expression, so the levels we find it in a cell, or how it functions. So these are the therapies that have been slowly entering into the field. But one thing that we do need at this stage, when we are talking about therapies targeting human genes, is to also talk a bit about mouse models and their utility. So mouse models obviously have brought us a long way, and we know that there are many things that mice do well. But one thing that mice don't do well is that they don't express a human gene. So we need human genes in order to test our gene therapies. We also want to know more in terms of disease mechanisms, what happens actually in brain development, and we also need all those genes that are expressed in different way in human brain compared to mouse. That's why I'm introducing to you induced pluripotent stem cells. So these are cells that can actually be obtained from each of us here in the room. We can get a little skin biopsy or a blood sample and use those cells to transform them or make them forget that they were a skin cell or a blood cell and give them an opportunity of a new life. So in case of studying epilepsy, those would be some brain cells or some brain organoids. But we can equally produce heart cell or muscle cell, so anything. iPS cells can produce anything that we want to study. The idea here is that our studies will then lead into or go back to patient and provide benefits for patients. So the two main things we can actually achieve by studying these stem cell or iPS-derived models is to understand the disease origin, so to learn when does the disease start, and also how it affects the brain development, because we have already mentioned these children have developmental delays, so many other things are changed, not only epilepsy. And the second utility is to enable precision treatments, which means maybe we uncover some other targets that are not exactly this gene, but that are changed in more than one patient with DEE. But as I already mentioned, the most important use at this stage would be also to use them as predictive tools for the drug efficacy and especially gene therapy efficacy. So I will give you 3 short examples of this, starting with SYNGAP1 DEE. So the girls you see on the right are girls that have SYNGAP1 DEE. Their names are Jaylee and Dali. They're from Melbourne, and they have all the things listed here and have been struggling with those practically from birth on. So you see that this does include epilepsy, but many other conditions that are difficult or not easy to manage. SYNGAP1 is one of the proteins that actually organizes how the nerve cell responds to the information it receives from other cells. So it's obviously a very important gene. And also from my analogy there, we know that it actually loses function, so it actually doesn't work enough. So what we did is to take Jaylee's cells and turn them into induced pluripotent stem cells, and then make these neurons that you see here in green. They look pretty nice. And then we also studied what are the differences between those cells and cells that are derived from somebody who is healthy. And we see all these bubbles in the middle just tell us that there are many genes or gene expressions, so there are many things that happen in these cells that are different between Jaylee's and control cells. Also, we can see the blue and green line. The green line actually represents how much activity we see in Jaylee's cells, and that actually tells us that even though these neurons don't even talk to each other, they just look like neurons, they still have changed activity or increased activity in Jaylee's cells. So we already learned from this that SYNGAP does start much earlier than what we see when patients are born and present in the clinic. That's why we went to use this model. This is called neural rosette model, and it actually models the brain development, human brain development within the first month of development. So 1 month after conception, the brain is kind of giving us structures that resemble these. And what you can see here on your left, in healthy neural rosettes, you see a really nice symmetric structure. You see a very well-defined little circle in the middle. We call it lumen. And we kind of know that these are good-looking rosettes. So everything looks okay with the way these brains would be developing. But if we take Jaylee's cells and make the same model, then we see that there are some changes there. This lumen, or this circle in the middle, looks wider and it's not so striking. And we know that this could indicate that actually the disease already starts so early in neurogenesis or in brain development. Now, quite strikingly, we used one of the ASOs that targets specifically SYNGAP1, and it fixes that lazy protein, and that does make this neural rosette look more like control, I guess. We all see that, which tells us also that this effect that we see in Jaylee's cells is also mediated by SYNGAP1, because we manipulate how much of SYNGAP1 we have there. So if we put more of SYNGAP1 in Jaylee's cells, they start to look more like healthy rosettes. And this is also useful for the screen of other drugs and treatments, because it tells us that simply based on morphology we can identify what could work in a patient. Now from SYNGAP1D, we're going to one of the common ones, or one of probably most known D forms, and that is Dravet syndrome. Dravet syndrome is caused by mutation in the sodium channel, so the mutation in the channel that regulates how much activity nerve cells have. And we know that this is 1 in 40,000 globally, because of the large number of patients, this is a syndrome where there is a lot happening in terms of drug development, and there is even ASO. There was the first ASO that was tested in human for DEE. And what we did is to get cells from a Dravet patient and make brain organoids. So you see here that within 2 months, these little clumps of cells turn into some ball structures that are 1 to 2 millimeters large. And those we can study in many different ways. But what I'm just going to show you here is that if you look at healthy cell on the left, you see that there's these spikes, which means there's some action potential. So these cells are active. But if you look at Dravet cell on the right, there is not much going on there. So this was telling us that we can see what is happening in a patient's cell line, and we can understand how the sodium channel being lazy again or not working well can affect the activity of neurons. The thing that I do want to show is that we can also show in this model that the drug that is currently in use in Dravet syndrome and shows really promising results, and Katherine may tell more about that, looks actually really good. So if you look on the left, there is some lightning. So we are looking at the activity of cells in these organoids or mini balls, and you see that there is some activity there, some flashes of light that shows when the cell is active. But if we put fenfluramine on the patient organoid, you will see that there is much more activity, especially towards the end, as it usually is. Yes, so I hope everybody picked that. So that tells us that this model can actually tell us what the drug will do in the clinic. So it can be predictive for the efficacy of drug for the patient treatment. So finishing with Florey's success story, so the SCN2A, another sodium channel, another very severe epilepsy or a spectrum of epilepsies caused by this gene. And the work at the Florey started with gene therapy for SCN2A probably 8 or 9 years ago when Steve Petru, who was leading the project, has established connections with Ionis and later also Praxis, and started doing work on antisense oligonucleotides for SCN2A. So the study that was done at the Florey was to look at the mice who carry this overachieving SCN2A. So SCN2A, this gene can both work more and work less, and if we focus on those that work more, they actually cause epilepsy earlier in life. We can use an ASO to remove the protein or reduce the protein. So you see here that ASO would bind to the mRNA or that messenger RNA and destroy it, which means there is less of SCN2A. And that is helpful because in the lines you see below, you can see that it actually increases survival of the mice that carry such mutations. So the orange line tells you that if the mice are not treated with ASO, they die within 1 month, but if they are treated, their life is significantly prolonged. Now, I have also mentioned that mice genes and human genes are different. So, of course, in order to translate something to the human or to a patient, we need to have a human model. And this is just showing you that ASO that would treat patients or treat humans was actually tested in brain organoids and could also show that reduction of the sodium channel expression, which can predict the efficacy in patients. So where did this lead further? This drug or this down-regulating ASO is now called elsunersen, and it is in clinical trials. The first part of the trials have already been completed last year, and those were safety studies showing that everything is going well, but what was really striking is that even with the low doses that were used last year and also a small number of patients, there's a 40% reduction in seizures in these patients, so that's quite promising. And you can see here that there is a clinical trial ongoing this year. So I guess what Chris and I want to tell you, and I think Katherine will add more flavor to this, that the standard of care at this moment with somebody coming to the clinic and presenting with seizures and probably being diagnosed with DEE is that they will get their genetic diagnostics and be classified in one of the versions of DEE. There will be some studies that tell, and some indications that tell clinicians how to treat them, but basically majority of drugs that are in use are already available, anti-seizure medications, which means that they can help to improve seizures. They can sometimes worsen seizures, as Chris said, or they have no impact, and especially they have no impact on developmental delay and all the other things that are happening in these children. What we foresee or try to work towards for future is to add to this genetics also the models. I would say stem cell models, Chris would say mouse models, but also gene therapies. And I see integration of AI and data analysis, so that once that patient presents, after some analysis and some work being done, they will have their targeted treatment, something that can improve both seizures and also the other outcomes. So that would be it from me. I just want to thank all the people who worked, obviously, on different projects here, and give a shout out. I don't know if there are many GETA people online, but GETA is Genetic Epilepsy Team Australia, a parent group of children with DEE, and we have a wonderful collaboration with them. And you will see Stu also later on the panel. Thanks to him for supporting our work, and also to Danielle and Dan Williams. So thank you very much.
Thank you very much, Snezana. So what we heard there is really going from science fiction to science fact, taking skin samples of patients affected by these complex genetic disorders, growing them into a mini brain in a dish, and then using state-of-the-art gene technologies to turn that around, and then going full cycle from work done by Snezana and team and Steve Petru into developing new drugs which are going into clinical trials, which is really, really extraordinary, and something that we really must celebrate, that innovation here is leading to treatments that will ultimately change the lives of these children and their families. So I think that's extraordinary work, Snezana, thank you. So next in our superb lineup of speakers is Dr. Katherine Howell. Katherine is a neurologist and epileptologist at the Royal Children's Hospital, but she's also an accomplished researcher in her own right. She's Neuroscience Group Leader at the Murdoch Children's Research Institute. Now, clinician researchers are hugely valued in our sector because they ensure that patient perspectives remain central to the research that we do. So please welcome Katherine.
Thank you, Peter, and thanks everyone for having me here today to, I guess, give a little bit of a clinical perspective and highlight exactly what the kind of work that Chris and Snez and teams are doing, how that impacts on the children that we see in clinic. So I don't need to remind you of this. I think Chris and Snez have illustrated it really well, but there's an enormous unmet need in these conditions, and they affect a lot of people. They don't just affect the person that has it, they affect their family, they affect, these conditions are really pervasive, I guess. And you've heard that there's over 1,000 different genetic causes. Majority of the causes are genetic. You've heard that they're often treatment resistant, and you've heard, and we're increasingly understanding, that the treatments that work best for one condition don't necessarily work best for another condition. In fact, they may make it worse. And essentially, to know how best to treat someone, we need to start with knowing the cause. So precision medicine, if that's our goal, if our goal is to give the right treatment to the right person at the right time, we need to start with diagnosing someone. What is the cause of their condition? And ideally in these conditions, we want to be able to do this really promptly because you will have seen that developmental problems are a part of this. And when you've got uncontrolled seizures in the developing brain, that adds to the developmental problem. So there's a real imperative to get the treatment right as early as possible. So we need to get that right so that we can then give the kinds of precision treatments that transform outcomes. And what we ideally want is a treatment that can address the underlying cause, can address all the symptoms of the condition, and Snez gave some examples of that. But we also really need treatments that actually target the specific symptoms in the right way for that person. And Chris gave some good examples of the HCN1 mouse and how that's impacted seizures. And for a number of conditions, we've got examples of treatments that really improve seizure control. What we don't yet have, in fact there are no evidence-based treatments yet, is specific treatments for the other symptoms of these conditions, for movement disorders, for sleep problems, for behavioural problems, things like that. We've got a lot of treatments we can use. We try them. We try, we fail, we try again. And that's not the approach that we want to be able to take. We want to be able to do this better. And the kind of models that Chris in particular was talking about are a great model for us to be able to start looking at treatments for the other symptoms as well. But if we come back to the clinic, we can't implement the right treatment till we know what's going on. And so we had a really big problem, in fact we still do in most of the world, with doing this the best way we can. We've got lots of different causes, we've got lots of different genetic tests that we call genomic tests, where we can test all of everybody's 20,000 genes at once to try and find a cause. We know they have really high yield in these conditions, we know that they're cost effective to use, but still in clinical practice we have a lot of trouble accessing them. Accessing them can often take many years. We often do it sequentially, do not the Rolls-Royce test first up, so we have to keep going. And that delays diagnoses by months, sometimes years, and that delays institution of the best management. So we want to change this. We want to say what would happen if we did the best test straight up when someone starts having seizures, and we do it quickly. And from that came a study that we've been doing called Gene Steps, which is part of the International Precision Child Health Partnership, which is a consortium of world-leading paediatric research institutes, so Boston Children's, Toronto Sick Kids, Great Ormond Street in London, and us over at Melbourne Children's, the Royal Children's Hospital, and Murdoch Children's Research Institute. And this was a study of rapid genome sequencing in infants with epilepsy, and most of those have a DEE, where the cause was unknown. So at presentation with seizures, they would get the best genetic test we have, which is called trio genome sequencing. We'd do it quickly, so 3 weeks turnaround time instead of many months. And what we showed is that we could actually do this in the clinic. In fact, this was the first time this had been used outside of an ICU in any condition, the rapidity of the testing. So we could do it. It had high diagnostic yield, over 40% got a diagnosis. In fact, we're up to close to 50% now, which is great. And the really important thing was the clinical impact. So essentially everybody who got a genetic diagnosis had at least one impact on their management in the short term. And for over half of them, one of those impacts was specifically changing their treatment. And so we're now, the cohort when we published that was at 100 big, we're now over 400. It's very much changed our clinical practice. We're just about to submit an application to Medicare Services to try and get funding for this so it can be rolled out across the country. And so we're essentially getting to the point where we hope we'll be able to do this for all people. And so what does this actually mean? So if we just look at one specific child who had this, so this is a boy called Levi. He started having seizures when he was 1 day old. He was quite floppy, and because of the seizures and how floppy he was needing help with his breathing. He couldn't feed, he needed a feeding tube in. We gave a number of different treatments. They didn't work, he was still having seizures. And so he had rapid genome sequencing, which showed he had a particular condition called KCNQ2-DEE, which is due to a potassium channel gene. Now we know that has a specific treatment implication, so we were able to start him on the right seizure medicine, take him off all of the wrong seizure medicines, And since then, he's had only a handful of seizures on one drug at pretty low dose, which is really game-changing compared to what usually happens for these children. He's now about 4. This photo on the right's a little bit old now, but most children that have a condition of the severity that he had early on never sit up, let alone walk. He was able to walk by the time he was 2. And when I saw him in clinic last time, he came in and showed me some karate moves. So his outcome, I think, was really very different to what we would have historically expected. Now, this absolutely is far from perfect. There are still a lot of additional problems that he has, including a lot of developmental problems. We've got a long way to go, but this is a really big step. And so if we look at the goals that we have, we come back to those. With this kind of thing, we can promptly diagnose people, and if there are treatment implications, we can implement them early and promptly. And in most cases, seizures is where we're making a difference. And there's still a lot of cases where we can't make that difference and we need more work, but we're starting to get there. Where we are, I guess, really sort of just on the cusp of making really big changes, and where the Florey's work is going to be so impactful is in the rest of the aspects of precision treatments. So things targeted the underlying cause, the ASOs that Snez mentioned, gene therapies, and we actually have, over at our institute, started doing some gene therapies for genetic epilepsies last year, which has been, is amazing. And so we're equipped to implement the kind of treatments that come out of the Florey and be able to advance things for the children that we see, which is amazing. Then we've got all other features of the conditions, and one of the things that Chris didn't really talk about, I guess, is within the package of work that they've done on the HCN1 mouse. You can see, I'm just going to give you some examples of HCN1 on the left-hand side and SCN2A on the right, so I'll just talk you through them. With the all of the work and the mouse model that Chris and team have looked at, they've not just investigated seizures, but also other aspects of things. They've published on changes to vision, published on changes to sleep. And this is the kind of model that can be really comprehensive and look at all features of this condition. And that there's now a pipeline that can be used for lots of different genes and be much more efficient in making an impact. On the right-hand side is the SCN2A stuff that Snez talked about, and I think this is another thing that also highlights the importance of the clinical and lab partnerships that we have had over the years. We've all worked quite closely together for some time, and in the clinical side of things from the SCN2A point of view, our studies there and delineating the different symptoms of the different subtypes and things were useful, I think, to these guys in the development of treatments and things, and we've sort of now got the kinds of cohorts of patients we can test these treatments on. And so they've been really fruitful collaborations, taking us from the bedside to the bench to the bedside, as we like to say. I think it's probably better words we could use, but that's how we like to do it. And this is a body of work scientifically, but the kind of thing that I can say we really value this kind of work because it makes a difference to the people that we see. And that's people like Ebony and Fern with HCN1, and people like Will and Eva with SCN2A, and really comes back to we're doing this for people. And so now we have this amazing pipeline of work we can do. All the collaborations are really scaling up so that we can do this from go to woe for newly diagnosed people, be able to make a change through all of the body of research and deliver precision medicines to people. And I guess really, this is really a game changer, I think, with where the Florey is going. So it's wonderful to be able to be a part of that work. So thank you to all. And I haven't got an acknowledgement slide, but I will say that the clinical side of things, I'm one of many. We have a huge amount of collaborations, and in particular, you might have heard of people like Professor Ingrid Sheffer and Sam Berkovic, who are really integral to this kind of work, and also have long-standing collaborations with the Florey, and also obviously to all of the patients and families that we look after. Thank you.
Thank you very much, Katherine. It's extraordinary impact and just such tangible impact. The impact of an accurate and timely diagnosis must be profound for these families, combating the uncertainty that I think would have been the experience of Mel and Stu. So, really impactful research and the rate of change, I think, is extraordinary to me. The fact that we're now delivering gene therapies in the clinic is really exciting and something that we can all really look forward to what the next few years hold. So we're now on to the panel session. So I might invite our speakers to take a seat, and also joining our speakers, who are Chris, Snez and Katherine, are Stu Place and Mel Anderson. So please take a seat, and while you're taking a seat I might introduce Stu and Mel. So Stu's son Will, aged 4, has SLC 6A1 DEE, and Mel's daughter, aged 17, has Pura syndrome. And Stu and Mel are both united in their commitment to make a difference for their children's future, and have done so in the form of really visionary fundraising, as well as bringing together a community of support. So we look forward to hearing about that. But Stu, I might start with you. So I was really fortunate to meet you as you were planning a superhuman challenge of riding 15,000 kilometres over 50 days. Not only did you succeed in achieving this extraordinary goal, but your story really touched the hearts of so many people. Can you tell us a little bit about your son Will and how he's motivated you to raise funds for epilepsy research?
Yeah, thanks, Peter. It's great to be here. My youngest son, Will, who's now 4 years old, he was first diagnosed, I'll consider our family as one of the lucky ones, quite early. So probably around 12 months of age, using our eldest son as a benchmark in terms of developmental milestones, we knew that he was lacking a lot of developmental areas in his development. So we, as parents, we had that parental instinct that something was wrong, despite family, friends and others telling us that everyone develops at different rates. We were very lucky that our paediatrician recommended a genetic test that eventually uncovered the mutation of the SLC 6A1 gene. And that allowed us to obviously implement, in early intervention strategies, or to help with his developmental delays and obviously get on top of his seizures. The way that the disease impacts him is obviously he has myoclonic atonic seizures. When he first started having them, he was having up to 150 a day. We're very lucky we're on top of that now. So we're really focused on making sure that remains the case. He has a mild to severe intellectual disability, mild to severe autism. He's completely non-verbal, which brings its own challenges. He has sleep, behavioural, movement disorders. So many of the morbidities that we've heard about from the speakers today. When we got the diagnosis, as a family, when you're battling with rare disease, you feel alone and you don't really know what to do. So you have to really build your own and create your own path, and that's what we did. So we knew we had to fight for our little man, Will, and I think there's a picture of him up there. There he is. And so that's what really has driven me to obviously try and work on his developmental aspects, and keep on top of the seizures, but also start understanding and researching genetic-based therapies on what can eventually, hopefully, help him in the long run.
Thanks, Stu. It's really extraordinary, the lengths that you've gone to and the effectiveness of your outreach. You've raised so much awareness as well as funds, and I'll just divert your attention to the big cheque in the background there. That's the fruits of Stu's efforts. He's generated over $385,000 to date, which will go to support research into Will's condition here at the Florey. So put your hands together for Stu.
Thanks, guys. Thank you.
So moving from one superstar parent to another, Mel, your daughter Sarah was the first person in the world, I believe, to be diagnosed with Pura syndrome. And you started the Pura Foundation and have made significant donations to medical research as well as raised tremendous awareness. So can you please share Sarah's story with us?
Hello, yes it is. Thank you. So my name is Mel Anderson. I'm not sure if we're getting that is my Pura Perfect Sarah. So Sarah was the first Australian diagnosed with Pura syndrome back in 2014. She was one of 4 that appeared in the first medical paper published in October 2014 on the 23rd, and that day became Pura Syndrome Awareness Day for us globally. I was privileged to be part of the Global Foundation. I was a founding board member in our global group. I stepped off that board after about 5 years and said to everyone here in Australia, once we reach 20 cases, then I'll look at doing something in Australia. My little Sarah was born, and very quickly we worked out that something wasn't quite right. Her temperature plummeted to 32 degrees. She was struggling to breathe. She had no suck reflex, no gag reflex. She needed feeding tubes. Everything just wasn't quite right, but no one could tell us why. Our diagnostic odyssey took 6.5 years because at that point, genetic testing just wasn't an option. So the doctors were testing one condition after the other, after the other, after the other. At 14 months, Sarah suddenly started putting her hands in the air, and I thought, oh, yay, she had physical disability. That was what we were told we would be dealing with. So at the time, I thought, this is great. Maybe she's progressing. Maybe she's done something wonderful. However, when we met the doctors a couple of days later, our neurologist was looking slightly horrified. An EEG showed that Sarah had a hypsarrhythmic pattern. She had infantile spasms, and we were told she would seize likely every day for the rest of her life. At her worst, her seizures got up to about 100 a day. Her longest seizure went for about 20 minutes, and the rescue meds that we were told would help did very little to stop it. Her strongest seizure broke her leg. So over time, the relentless and repetitive nature of these seizures meant she'd developed a hairline fracture through her femur. And one particularly strong seizure actually broke through that leg. So we ended up in the Royal Children's in traction for 2 days before she went back into surgery to have pins and plates put in her leg. So I think one of the things that I'm acutely aware of is that epilepsy doesn't just affect the brain. It's far more than that. And as Katherine touched on, there are so many things that our kids are also dealing with along with the epilepsy. And as carers, Stuart and myself deal with so much watching our kids go through this and feeling incredibly helpless because there's nothing that's working. In those first 7 years, we tend to use sort of 7 as a marker for Sarah because that's about the time of her diagnosis. We failed 20 anticonvulsants, as Chris touched on, and many of those meds actually increased her seizures. So we're being put on medication to try and stop this activity happening, and we're watching them increase. And if they didn't increase, often they just changed. So we went from having drop seizures where she was completely losing tone, to jerking seizures, to tonic seizures, to absent seizures. The hardest were the photosensitive seizures, where we had to live in the dark for a week because lights like these, or taking her out into sunlight, triggered her epilepsy, and she would just start seizing uncontrollably. So we had to live in the dark while her neurologist tried to work out what meds needed to come in, what needed to come out. It's that roulette wheel that Chris is talking about. And our neurology teams are doing the best they can, but nothing's working. And the hardest part for me now, as somebody that's the head of a foundation, is watching kids get diagnosed and they're still playing that game of roulette because we have no other options. So I'm watching them fail the same meds my daughter failed 15 years ago. And we need to do better, but to do that, we need a new way forward. And that's why we're here with Florey, and that's what led us to Chris's team.
Thanks, Mel. It's an extraordinary account of what must be just such a difficult journey to have endured, and to have done so with such grace is, huge credit to you and your supports. Chris, that must be incredibly motivating to know Mel and Sarah's journey for the team. Can you tell us how interacting with patients and families influences the work that you do?
All right. It's a tricky one. It's hugely motivating, but it's also incredibly frightening. You want to help, and we have help, in some ways. I don't know what we're going to do for Pura. I can't promise anything, but we're going to try. Being a fundamental scientist, you don't get the exposure that Katherine gets, and it's been humbling to say the least. but motivating. And I mean, we're hoping that deep care comes together, if not through a frontiers, through some mechanism, so that we can potentially help as many people as we can. You know, not just the Pura families, but so many others that are going through the same journey.
Thanks, Chris. Snezana, I'd like to just pick up on something that was embedded in your presentation, in that the genetic therapies that you and others at the Florey and elsewhere are working on are really looking to tackle the underlying cause of the disease. And I think it's what Katherine alluded to, about this offering hope for some of the other features of the disease. Can you give me your perspectives on that, or perhaps correct me if I'm wrong there? Do you have hope that these genetic therapies might have greater influence than just controlling seizures?
Yeah, that would obviously be the goal, because having anti-epileptic drugs, improving anti-epileptic drugs could potentially, and doing a lot of screening in mouse models could potentially identify controlling seizure one. But what we hope is that if you remove the cause of disease, that you will also remove the challenges that the organism has in any aspects. Now, of course, there is a problem there. Of course, we are aware of that. That is that we are intervening and trying to fix something that has been broken for many years by the time these children will hopefully receive gene therapies. So how much of that is reversible will, of course, be something we have to see. However, I think everybody agrees having a bad or rotten apple removed from the system shouldn't be damaging. So it probably can only help. And we hope for some genes, it has been shown that there is hope that many other aspects of disease could be solved if the therapy is applied later in life.
Fantastic. Katherine, I wonder if I can ask you to cast your eye to the horizon. Where do you reckon we are going to be in 10 years' time, given what you've seen and the rate of change and work from Chris and Snez and others? Where are we going to be in 10 years? Are we going to hold you to this in 10 years' time?
Can I go big?
Yeah, please.
Yeah. What we would like to see is that when children present with seizures and with one of these conditions, that we quickly find the cause, and that we're then able to say, well, this is whole body of work showing these treatments are the best, and we will implement them. And that includes disease-modifying treatments that improve the underlying cause. And we're starting to get some of those, including clinical trials for the most common of the genetic epilepsies, Dravet syndrome, that Snez alluded to. And actually what we hope would be that we could give one of those therapies, assuming that they work, pretty much straight after diagnosis. And for that particular condition, that's at about age 6 months, when they start having seizures. And children with Dravet syndrome are typically developing around the time they start having seizures. They are until sometime usually in the second year of life. And ultimately they end up usually with a moderate to severe intellectual disability and a very drug-resistant epilepsy. But Snez was talking about, well, when is too early or too late? I mean, you would think that actually if you could give them before development starts to become abnormal, that actually could be the perfect kind of intervention and the perfect time to intervene, and we would love to be able to have a treatment that could do that. That's obviously not the case for all conditions, and sometimes the developmental and other problems predate seizures. And we don't know what is the case there, but I would hope that, I would love to say for all of the conditions we could do something like that. And I think ultimately that's probably not realistic when we're dealing with 1,000 different conditions. But I would like to say that for at least some of them we could do that. And I think we need to be strategic, we need to be efficient, and some of the types of things happening through here I think are really exciting. It's great to be able to be part of them.
Fantastic. Now, our panellists have very generously opened themselves up to questions from the floor. I know we've got a couple online, but do we have any questions from anyone in the room? I think we've got a roving mic.
I'd like to grab someone.
Yeah, grab mine, because I don't need to answer.
Hi, a very naive question. The genes that you're working with to control seizures, is that all the work that they do, or are they applicable or do work in other reactions and conditions of the body? I don't know who I'm asking that of.
Well, I can maybe go. So the DEE, so these early onset epilepsies, actually overlap quite a lot with other neurodevelopmental disorders like autism, and we've seen also some of these genes are linked to intellectual disability. So we know that disorders that are linked to development, so where we maybe not necessarily see seizures, but see autism, intellectual disability, developmental delay, they could profit from some of the therapies that we are developing, and where we can look into, improving the conditions or improving their lives too.
Chris, just to continue on from that point, I understand with the proposal for the Big Frontiers grant that you're interrogating many different genes and building the mice, but then you're going to bring that all together with computational biology to make sure that we can identify new pathways to inform a broader array, more than just the 20 mice that you're looking at. Could you just add a little bit of detail to that?
Try. I think we are going to find common hubs. We have 1,000 genetic causes. We already know that there's causes that you could lump together. As we do that, our understanding of how those epilepsies occur will inform not just the rare epilepsies that we're talking about now, but the more common epilepsies moving forward. And I think Snez has argued quite well as well. It won't just be epilepsy. There are things that we're going to learn from these different model systems that are going to tell us about the autism, about potentially other diseases even in the eye, and some of the stuff that we're doing. So I guess the answer to your question is what we're doing is starting relatively small, but the impact could be vast.
Great, thank you. Any other questions?
I think, Katherine, this is for you. We've heard a lot about comorbidities, epilepsy and a lot more. I'm thinking about a very young child with 100 seizures a day. What's the morbidity of hippocampal sclerosis?
We're talking about there is that the hippocampus is a structure in the brain that's involved in in memory, and seizures can actually damage that and cause scarring on it, which can then cause epilepsy later in life. So just as an explainer of the question there, I think the answer is we don't know entirely, because in a lot of these conditions people keep having seizures and we assume it's the same cause and they don't necessarily get another MRI scan. So we don't know that for sure. But it actually is probably lower than what we expect, and we don't understand the reason for that.
We might go to a couple of the online questions. So for that honour, I'll turn to Georgia.
This one is partly answered, I think, by yourself, Katherine, in your presentation. But are there efforts to improve developmental outcomes in KCNQ2-DEE, as an example, beyond seizure control? What treatment strategies are being explored to promote neurodevelopment and address the underlying developmental challenges in affected individuals?
I don't know, do either of you want to take that in terms of anything you're doing in the lab?
For KCNQ2, we are developing IPS lines. We see the developmental problems where they could come from. So there's delayed development of neurons, and there are changes or important role that this channel does play early in development. I think that you illustrated, Katherine, that the stopping the seizures could help already. So I think that's probably a really good approach, and that's kind of also surprising for me. I didn't know that works, because there is a lot of speculation whether stopping seizures in some cases would actually resolve the developmental outcomes. But it seems if it helps, that's great news.
Yeah, I mean, it definitely doesn't resolve them, but I think there's ways in which it probably does improve them. Yeah, I think there are, there's a trial of a particular small molecule that is targeted at the channel, which is something that's sort of potentially could help. At the moment, we're very limited to doing the best we can for seizures, reducing the burden of medications and impact on those, improving someone's general health, and early intervention strategies. That's our package at the moment, and we need more.
I should say something. I mean, it's not just about the children, it's about the carers as well. You know, horrible sleep deprivation, you know, the dealing of it's a family impact is extraordinary. So I think we are focused on the children, and we should be, but the impact is well beyond that.
And it goes beyond that too, because that affects our workplaces, it affects our families, it affects our medical practitioners. When you're standing in front of your child's neurologist and they're giving you the same blank look you're giving them because they've run out of options, that also affects their mental health. So people forget that mental health level in the entire team. I mean, we're not just talking about families. This is foundations, families, clinicians and scientists all being impacted. But at the same time, we're all able to work together. And that's where our power lies.
Did you want me to go with another one? Yeah, there is
one more online question.
There is a great one, and I think you, Katherine, again, you might have touched on this with the 6 months. But is there an optimal time window for administering ASOs or gene therapies, such as before the age of 2, to maximise effectiveness?
I think we actually don't know, and it will almost certainly vary depending on the condition and its manifestations. We have a lot to learn.
I think it's only fitting that we end this panel discussion with some reflections from Stu and Mel. I'd love to hear how important it is for you to have a community of support, other parents similarly, you know, with kids similarly affected. How important has that been for your journey, and are there any other things that you'd like to share with the audience?
You would have seen in Chris's presentation, there was a photo of Chris with us at the Melbourne Marathon last year. So we're very big on community. We formed our foundation October 2023. So we've been going just over a year and a half almost. And that was because we had enough numbers with our families to bring everyone together. Being rare in Australia means we are spread out. So we have families all over the country and in New Zealand, because we also support our New Zealand counterparts. And coming together gave us a voice, because that's often the thing that's lacking for families when you're rare. When you're dealing with epilepsy and your child is having 100 seizures a day, the first thing you do is withdraw, because you're struggling to get through every day. So being able to have that community there and being able to say, okay, we haven't heard from that mum or dad for a while, checking in, are you okay? What can we do? How can we help? But at the same time for them, what gets them through some days is these guys, because they know someone's out there trying to make that change. We're living a cycle that we can't break. If we always do what we've always done, we'll always get what we've always got. We have to change. And it was that change that Chris is talking about, that he's been working through, that led us here. And it's that change that gives hope to our families. When we came together at the Melbourne Marathon last year, we had clinicians from the Murdoch, we had scientists from the University of Melbourne and from the Florey Institute. We had families, work colleagues. It was a giant group. I think we had 5 families represented and 96 people in attendance. When you're rare and you turn up at an event, and Miss Jenna over there is highlighting it for us, in one of our purple tees with Pura written on it, that was the first time some of our families had ever met another Pura patient. And for them to actually come together and meet one another is an incredibly powerful thing. And for us as families and as organisers of these foundations, that's why we do it, is for our families, to provide support, to increase capacity for research, but also to try and give hope to our families for a new future.
Yeah, I think you've nailed everything there, Mel. I'm not sure I can really add to that. Yeah, look, much like Mel and the Pura Foundation, you know, myself or our family and 2 other Australian-based families created a foundation, SLC 6A1 Kids. You know, first and foremost, we wanted to connect the Australian community, which we're starting to do. We're probably not doing a good enough job, because we're volunteers and we're working full time, and we're trying to learn and educate ourselves and others about the disease and fundraise and everything else. So there's still a lot of work to be done for our particular gene mutation, but the foundation is a great starting point. We've connected roughly about 15 families in Australia. We know of around 15 or 16 diagnosed families in this condition locally, and there's much like your disease, the Pure Foundation. We've probably got about 600 or 700 diagnosed families that we know of globally. So, yeah, we just want to be able to share, what works, what doesn't work, support each other with the highs and the lows, and then work with scientists, researchers, and institutions like the Florey to really move the needle on advancing the genetic therapies that we need for their future.
Thanks so much. Please join me in thanking our panellists. So just to end, I'd like to thank you all for taking the time to come and learn about the extraordinary work being done by the scientists here and our clinical collaborators, and to hear these extraordinary perspectives from parents of children affected. If you feel inspired by the research that we're doing into genetic epilepsy, please consider making a donation to the Florey. The generosity of donors is really vitally important to making this research possible. And should you have an interest in making a donation, please reach out to any Florey staff and we can direct you to the right people. I'd like to just reiterate my thanks to you for joining the first Florey public lecture for 2025. We've got many other events lined up this year and we'd warmly welcome your attendance. So in closing the formal proceedings, I'd invite you to make your way out to the foyer where there's some refreshments and the chance to have a chat with some of the speakers. Thank you.